go ahead and get started. The first thing I'd like to say is it would be really nice if we'll move forward a little bit where nobody's contagious here, so it might make for a more congenial atmosphere. I'm really happy to be able to introduce Julie Christianson to come and speak with us from the University of Kansas. Julie and I have had many of the same mentors and strangely intermingled careers in some ways. She started at Kansas for a Ph.D. with her mentor, Doug Wright, who was a postdoctoral in my lab when I was a graduate student, and then came to do a postdoc at the University of Pittsburgh, where I was a postdoc, and then a research assistant professor, and then assistant professor. So Julie then became a research assistant professor, and in the pain center at the University of Pittsburgh, got tons of papers out and became very interested in visceral innervation. We both have been immersed in the primary afferent nociceptor field, but we wound up taking very different paths, and now she has gone back to Kansas for a faculty position, has just received tenure and extensive R01 funding, and is now going to be telling us about the intersection of pain with stress and maybe even some metabolic disease, and has even considered the possibility that there might be a role for the central nervous system in pain, which I'm not at all convinced of, but I'm excited to hear your talk.
Okay, thank you, Derek. It's a pleasure to be here. As Derek said, we've known each other a very long time and have very intertwined, overlapping family trees. So I'm also happy to be visiting from one idea state to another. I'm a big fan of all the idea programming, co-breeze, in-breeze, all of it. It's good for everyone. So thank you all for being here. So as Derek said, I was born and raised a primary afferentologist. I have kind of stepped away from that as I've gotten more interested in stress and the influence of life experience on how that influences how we perceive and process pain signals and during the lifespan. So I changed the title. Usually I have the title of my talk is Painful Consequences of Early Life Stress. That in my lab, looking at adult stress as well. So I've kind of shortened my title to make it a little more broad, but really I'm going to focus mostly on early life experience. So this is obviously a huge problem. There's a really nice study that was done, the ACE study. You may have heard people talking about your ACE score, ACEs, things like that. So these are adverse childhood experiences. So there's this big study done by Health and Human Services in 2012, doing a really broad survey of how prevalent this really is. And so in 2011, 27 out of 1,000 children were maltreated. And this is a steady increase over the 5 previous years. So it's a problem that's getting worse. There were almost 700,000 unique cases in 2011, and importantly, a quarter of these were in children under the age of 2. So this is a very pliable population. You obviously still have a lot of neurodevelopment going on. So this is a big problem. Interestingly, 2/3 of these reported at least one, meaning that more than one was occurring. So 22% reported 3 or more. So obviously, the more experiences you have, it's going to be an additive effect. And so there's an increased risk of health problems that increases with the number of ACEs that you experience. And they came up with this kind of pyramid showing that you have these experiences, which leads to cognitive emotional impairment, adoption of health risk behaviors, increase in disease, disability, and early death. So how do we fill in these scientific gaps that lead to one another? And that's kind of where we fit in. On the other side, so opposite of stress and abuse, we have Nicu admittance. So if you're born prematurely or you're born with complications, you're going to end up in the neonatal intensive care unit. And so neonates in the NICU will experience on average 16 procedures a day. 10 of these are considered to be painful. They're all considered to be stressful. This number increases if you're born less than 29 gestational weeks. And about 80% of these are performed without specific analgesic intervention. That's not because neonatal doctors are mean, nurses are mean to these babies. We just don't know how to treat them. So a lot is unknown. Up until the 80s, most of you probably know this, doctors thought that neonates didn't experience pain, which has obviously been debunked. So they do experience pain, we just don't know the best way to go about stopping it. And clearly, these babies undergo prolonged periods of maternal separation. So there are a lot of implications that can come from early life stress. Increased learning disabilities, increased mental illness, as well as functional pain disorders. So this can include migraine, fibromyalgia, and what my lab's primarily interested in, chronic pelvic pain. So what we're really interested in is how do you go from tiny baby in the NICU, a child who's undergoing stress and abuse, to have an adult woman who undergoes functional pelvic pain, and we're not gender specific, we're very inclusive in my lab, or a guy who's experiencing these symptoms as well. So a lot of this is housed in the regulation of the hypothalamic-pituitary-adrenal axis. So this is really the part of your brain that is going to respond to an acute stressor. So when a stressor occurs, there's a signal from the hypothalamus to release corticotropin-releasing factor to the anterior pituitary, causing releases ACTH to the adrenal cortex, releases glucocorticoids, which we all know are going to kill us all. Cortisol is the worst thing ever, apparently. Okay, so, and CRF also has peripheral effects on mast cells and enteric neurons that can cause a lot of systemic inflammation in the periphery. So under normal conditions, these glucocorticoids will feed back in a negative feedback loop, bind to receptors present at every stage of this axis, and once the stress is gone, it all normalizes. So this is what happens on a normal basis, but if you get disruption within this axis, obviously you're not going to have that appropriate feedback. Okay, so there are a couple of limbic structures that are involved. The ones we're primarily interested in are the amygdala and the hippocampus, which are also responsive to stress. But these have opposing forces onto the hypothalamus. So the amygdala really kind of drives forward this process. The more processing from the amygdala, more input there is, the more this activation will drive forward. The hippocampus works in opposition to this, performs a nice negative feedback onto it. And they also have feedback from the glucocorticoids to help keep the system under check. So there are a couple of ligands that I'll talk about later in my talk that are involved with this. Obviously, I've already mentioned CRF. There's also the urocortin family members, urocortin 1, 2, and 3. These work through a 2-receptor system, so there's CRF1 and there's CRF2. So CRF1 is the one that's been mostly shown to be anxiety driving. So if you agonize CRF1, you're going to get anxiety-like activities. You're also going to get kind of increased pain. CRF2 is more anxiolytic, so if you activate CRF2, it's going to decrease anxiety-like behaviors, and it's also going to antagonize CRF1 activation. Oh, and then we've got the glucocorticoid receptors. So there's GR and MR that will bind the glucocorticoids and also help quiet down this system. And CRF1 has been implicated in, at least in rodent studies, to be involved in increasing colonic motility and hypersensitivity. So this has been well studied in IBS-like models. And CRF2 has been implicated in stress-induced bladder, although I'm starting to question that a little bit from what we're doing. But it's also involved in visceral pain activation as well. Okay, so the way we do this in the lab, obviously we're not going to have little, babies from the NICU in our lab. That's just crazy. So we do this in a mouse. So we have a paradigm of maternal separation. So we order in pregnant dams during the 3rd week of gestation. And beginning on the day after birth, we will separate them, the pups en masse. We put them a little bit of the bedding in the bottom of a beaker, move the pups in, and they're held at 34 degrees, 50% humidity in an incubator for 3 hours a day for 21 days. And then we wean them at day 22 and they go on. And so we have a couple of different adult interventions that we've looked at. The first one is a water avoidance stress. We chose this as it's been shown to activate mast cells, cause mast cell degranulation. It's been widely studied in rodent models of IBS. So what we do is I went to Target essentially and I bought those little plastic 3-drawer bins. And so we took those and I hot glued a little Ziploc container on top of that. So now we have this little round platform. And what we can do is we fill this with water. And so we fill it with water until about like an inch below the platform, and then we pop a mouse on it for an hour. And this causes them a lot of anxiety, and it causes all their mast cells to granulate. It's pretty fantastic. So we can do this. So we have our separation protocol, and we let them hang out, and then we do a baseline measurement sometime during adulthood. We perform the water avoidance stress and then measure them again. So another intervention, this is kind of the opposite, whereas the water avoidance stress, we're trying to make it worse. We have a voluntary exercise intervention, and this hopefully potentially would make them better. So again, we do the separation during the neonatal period, and then at 4 weeks of age, we pair house them in a cage with a running wheel. We pair house them at this age because they're still very young. They've obviously already undergone maternal separation. These are social animals. They don't want to be singly caged. We're kind of worried about the extra effects that doing that. And at this age, they're still young, and they're with their litter mates anyway, and they'll run on the wheel together, like little running buddies. It's pretty cute. And so we'll let them run however long. Most of what I'm going to show is we started running them at 4 weeks, and we tested them at 8 weeks. And obviously, we're interested in pelvic organ sensitivity. We're also interested in comorbidities because patients that have pelvic pain disorders, they're more likely to have a second disorder, as well as mood disturbances like anxiety and depression. So we're really interested in kind of giving a whole body analysis of what their overlapping conditions are, as well as looking at gene expression within the HPA axis and those limbic regulated structures. Okay. So looking at pelvic organ sensitivity, most of what I'm going to show is looking at responses to pelvic organ distension. So what you can do, shown here, these are implanted electrodes into the abdominal muscles of a mouse. You can use these electrodes to measure the EMG recordings of the abdominal muscles. And so if you distend the pelvic organ, you get reflexive contraction of the abdominal muscles. And you can measure that, and it's termed the visceromotor response, or visceromotor reflex. And this is just the fancy diagram of all of the stuff we use to measure this. So what we can do, this is how we do colorectal distension. So we implant, if we do the chronic implantation, we do this 4 to 7 days before we do the recordings. When we instrument the amyl, we'll lightly anesthetize them, get them in their all hooked up to the instrumentation, let them acclimate for 30 minutes, and then we'll give a distension series. So there's a balloon in the hollow organs, or the bladder is catheterized because the bladder is a balloon. It's very handy. And then we can apply graded pressures and measure the visceromotor response during those distension periods. And what we end up with is something that looks like this. So this is increasing pressure, and this is, hopefully you can appreciate that the EMG activity in the muscles increases as we increase the pressure. And if we map this out as a % of the baseline activity that occurs prior to the distension, we get this nice stimulus response curve showing a physiological response to the increasing pressures. So all of the red female data came from the work of a very talented graduate student I had in my lab that graduated a few years ago, Angela Pierce. And so this is kind of our initial. I'm a big fan of the IDEA program because I was hired onto our COBRI. And so as part of my COBRI project was to look and see how this early life stress may affect vaginal and urogenital sensitivity as a potential model of vulvodynia in rodents. And so we're interested in vaginal sensitivity in these animals. And so at the time we were looking at 2 different paradigms for the separation. Most of the literature on maternal separation uses a 14-day separation. So we tried that as well as the 21-day. You can see we kind of got a dose response effect. So the 14-day had a bit of an effect. It was higher. So, sorry, I should say that. So the % of baseline EMG is on the y-axis, and then our balloon pressures are over here on the x. And naive is going to be in black. And these gray dots are mice that were separated for 14 days, and this is now adult recordings. So we saw a bit of an increase, but it wasn't significant. But when we looked at the mice that were separated for the full 21 days, we saw a huge significant increase in the EMG response. So as you can imagine, we've gone with the 21 days since then. And so further looking at the other organs, we also looked at bladder distension. So this is looking at responses to compressed nitrogen applied into the bladder. And again, we see a significant increase in the EMG response. The male mice, so all the male data, this is by another graduate student who just defended this May, Isabella Fuentes. This is her dissertation project. Male mice are not as easy to catheterize. In fact, I've never even tried it because I just think it's going to be grisly. So female mice are very easy to catheterize because of very short urethra. It's pretty easy. But the males, we can't as easily do bladder distension. So what we do is we apply graded von Frey monofilaments to the perigenital region and just look for withdrawal responses to application of the different filaments. Much like, I'm sure you've seen Derek show this data on hind paws and everybody else. And so what we see is a significant decrease in the withdrawal threshold, suggesting that there is mechanical allodynia in the perigenital region. Okay, so we've got vaginal, we've got bladder, we've got perigenital. And interestingly, when we looked at colorectal sensitivity, not only did we not see an increase, we saw in the females a significant decrease in colorectal sensitivity. And this actually made us feel a little bit better about the model, because if we just sensitized the entire mouse, that wouldn't have been as interesting. So this is, you know, it's a negative result. It's completely opposite of what the rat literature has shown. That's pretty much a singular colorectal increase in sensitivity and not urogenital. So it took a little convincing to get those folks to believe our results, but now they do, which is great. So we don't see it in the GI system. Okay, so I'm going to talk a little bit about, our mice are not this happy when we put them in a pool. The effect from water avoidance stress. So when we do this, so this is the area under the curve measurement, so it's taking that entire distension series and taking the area underneath that. At baseline, as I showed, we see an increase in this EMG response. Perform the water avoidance stress, we see this transient dip in responsiveness, but we see a rebound that's even higher than before. So we can exacerbate this bladder sensitivity by the water avoidance stress. And in the males, we run into a common problem with these von Frey monofilaments. They only go so light. So we've already bottomed out. They're already responding to almost the smallest filament we have. So we need to find something that's a little more sensitive to see if we can get there, but they're responding to even the lightest touch. So they're already sensitive and we can't really sensitize them further. Interestingly, when we looked at the colorectal sensitivity, we saw a little bit of difference. So in the female mice, one day after this water avoidance stress, we saw a significant increase in colorectal distension responses in the naive mice, but not in the separated. And we saw the opposite effect in the males. So one day after the water avoidance stress, we saw a significant increase in the separated and not in the naive. So there's obviously a sex-dependent difference in this susceptibility to water avoidance stress in our mice, and we haven't completely sussed that out. Okay, so now I'm going to talk about how voluntary exercise affects this. Back to the females in the urinary bladder. So in this case, the closed circles are going to be our sedentary mice. So these were weaned at 3 weeks, group housed, very little enrichment in the cage. And again, our separated mice show a significant increase in their visceromotor response to bladder distension that was not observed in 8-week-old mice that have been running for 4 weeks. So this introduction of the exercise wheel prevented the development of this, and this is just the area under the curve measurements over here. Same story with the males. So our sedentary mice had this significant decrease in their withdrawal thresholds. If we let them exercise, we don't see it. Okay, so we're also interested in functional output in these organs. So we have a very crude way of measuring micturition. We also have a very complicated and sophisticated cystometry system that is kind of a bear to use. So it's a whole lot easier to do this. So essentially we take a piece of filter paper, put it on the lab bench, put a mouse on it, overturn a cage bottom, and let them run around for an hour. And then we can expose the paper to UV, and we can count and measure the urine spots that are left behind. So when we do this, this is in the WAS study in the females, so we didn't really see any differences at baseline. There weren't any differences in the number of spots during the hour, or the total urine output, or even the fecal output. But one day after this water avoidance stress, we saw a significant increase in at least the micturition output. but not in the naives. We kind of saw the opposite in the males. So at baseline, we did see an increase in the number of spots, the amount of urine that came out, as well as the fecal output, which was actually diminished following exposure to water avoidance stress. And one thing I do want to mention is that these were repeated tests within the same group. So there wasn't a reintroduction problem with it, because it is a novel environment. So that wasn't an issue here. And these are separate groups, because we saw that decrease with repeated groups, and we thought, oh, they're just getting used to their environment. And we repeated it with separate groups of animals, and we saw the same diminishment. So we don't think this is just introducing them to the same environment causing a reduction. It's an actual effect of that water avoidance stress. Okay, so looking at the voluntary exercise in females, in this group we actually did see an increase in avoidances in our females at baseline that was not present in the exercised. And there was a difference in void size in the naive that were exercised, but not in the separated. And fecal output was increased, which you would expect because they're running and their system's working a little bit better than the sedentaries. Okay, so now moving on to comorbidities. So one of the big comorbidities in these patients is there's a high prevalence of depression. And one of the hallmarks of depression is anhedonia, or a lack of pleasure seeking. So you no longer want to do things that are pleasurable to you if you're depressed. So one thing that mice do find pleasurable is drinking sweet water. So we can give them a sucrose preference test. So we put them in these cages that look just like this, and they have 2 identical water bottles. And so for 24 hours, both bottles have water in them. And so we measure it, make sure they're not preferring one side over the other, and then we swap one of the bottles out for 1% sucrose. Leave them there. At the end of 24 hours, we weigh them, swap the sides, weigh them again. And then we can measure preference of sucrose. So when we do this, in our sedentary mice, we were kind of surprised that our separated females actually had a higher preference for the sucrose. And the exercise mice overall had a higher increase as well, which we could be considered to be a caloric effect as well from the exercise. But what was really interesting is when we looked at the sucrose preference, so sucrose preference is on the y-axis, the number of voids in that micturition test is on the x-axis. And so if we separate, our sedentary stressed mice fell into 2 categories. One category that kind of looks like normal, less than 5 voids over that hour, versus those that had more than 5. So when we look at those in the normal, both the sedentary and the exercise, there was a significant correlation between sucrose preference and the number of voids. So the more they liked the sucrose, the less they voided. So that kind of correlates increased anhedonia with increased output, kind of connecting those 2 together. In the males, we didn't really see a big impact of sucrose preference. If anything, exercise made them like it less, which is kind of bizarre. But we also saw the same, at least a trend towards a correlation where the sucrose preference is on the right, on the y-axis, and that perigenital threshold is over here. So the more sensitive their perigenital region was, the more allodynia they expressed, the less they liked the sucrose. Again, correlating that behavioral effect with depression scores. And finally, my last piece on this, exercise itself, running, is pleasurable to the mice. Some of you may disagree in this room. I find running pleasurable when I can do it and my knee doesn't hurt. But they want to run. If you put them in the wheel, what you can see here, so these are kilometers per day. They're running a 5K every day, if not more. They love it. They jump on and they run like crazy. But our separated mice, both the females and the males, run less. We don't think this is because it hurts. We don't think that they're actually like physically in pain and can't run. We don't think they're getting the pleasure out of it that a naive mouse does. So their reward system is dampened to where this running, they like it, but they don't like it as much as a normal mouse does. So this is another behavioral readout of the anhedonia. Okay, so moving on to the HP axis regulation. Again, just to kind of a reminder that the stress will affect these limbic structures, the amygdala as a feed forward, hippocampus as a brake. to this downstream activation. I'm just going to show hippocampal expression from the exercise studies in the females. This is kind of the cleanest, most straightforward data that we have on this. So CRF1, which again is that pro-anxiety in the hypothalamus, but in the hippocampus, binding of CRF1 would strengthen that negative regulation onto the hypothalamus. So this would be strengthening that negative regulation. And what we see is a significant decrease in our sedentary animals, in the stressed animals, both at sedentary and exercise, showing that they're losing this input. CRF2 was not affected, and it hasn't really been across the board in all of our studies. Glucocorticoid receptor, which again is going to bind that corticosterone and help regulate it, this is also decreased, again showing that it's not getting that negative feedback that it needs. But when we looked at BDNF expression, it's not really changed in the sedentary mice, but in the exercise mice, especially those stressed animals, it's through the roof. There's a lot of BDNF being expressed. And I'll get back to that, kind of my grand hypothesis statement at the end, what we think is going on. So another thing that I mentioned is the peripheral effects of CRF expression. So CRF is going to be released by the hypothalamus. It's going to go everywhere, and it is very involved in enteric neuron signaling and colonic motility and things like that. So one of the other things is that it activates mast cells. So mast cells have CRF inside of their granules. They also have CRF1 receptor expression. And so mast cells are exquisitely responsive to stress as well as to CRF release. And so we were interested to see if, and they're obviously involved in peripheral inflammation, neuroinflammation. So we wanted to look at the mast cell, just what they look like in these affected tissues. And so this is using toluidine blue staining in thin sections of the bladder over here. So this is naive bladder. You can see all these nice intact mast cells. And in the separated bladders, all of the mast cells are pretty much just exploded. All their granules are being released. So when we looked at this, we saw that at baseline, we saw a significant increase in mast cell degranulation. Water avoidance stress did increase degranulation in the naive. It didn't really have an impact in the separateds, but we think that's because they're pretty much already at peak degranulation at this point. We're also interested in looking at protein expression of receptors that are involved in the signaling pathway. So PAR2 is the tryptase receptor. So tryptase will be released by mast cells, can bind PAR2 on axonal endings in the periphery, which will then sensitize TRPA1. So this could be involved in increasing that pain sensitivity in the periphery. And we did see a significant impact of water avoidance stress, not of the separation. But at this 8-day time point, we saw a significant increase in both PAR2 and TRPA1 protein levels in the bladder. And just a bit on the male. So this is the male mice. This is looking at bladder and prostate. You can see again, the separated and the bladder have a significant increase in mast cell degranulation. And in the prostate, the naives were, so prostate has an increase in degranulation in the naives following the water avoidance stress. And it was increased in both of the separated populations. Okay, so what we would think is going on, so we would normally have this negative feedback loop going back up to the limbic structures. What we think is happening is basically the hippocampus in our model has gone offline. So we no longer have that negative break. It's allowed to go willy-nilly, and we have this increased activity of the HPA axis. When we come in and we let them exercise, we're not really changing what was happening. We're not fixing the problem because we aren't seeing that increase in CRF1 or GR expression, but we are getting this BDNF. And we're looking at doing experiments right now to look at BRDU labeling in the hippocampus to see if we do have neurogenesis going on. But we think we're strengthening those connections within the hippocampus to help reintroduce that break and help modulate the activity of the HPA axis. So some of the recent things that are going on in the lab, we're really interested. So this is work by a current graduate student, Olivia Eller-Smith, in conjunction with one of our colleagues, John Tifo. Olivia, if any of you saw my talk at Spring Pain, you know how I feel about migraine research. I don't like it because we failed at it for 2 years. So Olivia tried for 2 years to get a headache behavior in these mice because we looked at the dura, since migraine is a comorbidity with all of these things, if the dura had increased mast cell degranulation, and lo and behold, it does. So in the female mice, we get increased dural mast cell activation, exercise stops it, looks exactly like the bladder. We're like, cool, awesome, we can study migraine in these mice. Oh, hey, that's super hard. So one of the last things we did was inject them with nitroglycerin to invoke a migraine, a headache-like behavior. The experiment failed, but we had to weigh the mice to get the correct dosage. And when we did that, we noticed, oh, some of these mice are really fat. And lo and behold, it was our separated sedentary females. And so these are 10-month-old female mice. They're around 30 grams, which is pretty heavy for a female mouse. And the exercised weren't any different than the others. We're like, well, that's interesting. Okay. And John Tifo has an echo MRI, which you put a mouse in a tube, shove it in this machine, and it gives you back the body composition of the animal. So we did that and found that most of it was fat. Their lean body mass was pretty much the same as the others. It was all body fat. So these mice have about 25% body fat, where normal is around 15. So Olivia then dissected out the fat pads, saw periovarian, significantly heavier in the sedentary stressed, as well as the retroperitoneal fat pad. And we took it and we sectioned and had H&E staining done on the periovarian. And John told us, we're like, well, how do we measure this? He's like, just take pictures of it, put it on put it on PowerPoint, and you can see it. And we're like, well, that doesn't make any sense. We're like, oh, okay, never mind. Yeah, that's pretty obvious. So in our sedentaries, the adipocytes are significantly larger. He said, you can go in and measure them, but why? They're bigger. I'm like, okay, thanks, that's good. That kind of kills me as an anatomist, but sure. And so we're gonna chase after that. So Olivia came back and she had this group of males that were slated for migraine, and we're like, oh, we'll just change it to metabolism. So now we're looking at metabolic syndrome. So these are 3-month-old male mice. We aren't really seeing significant differences within the sedentary mice yet, but we do see an effect of the exercise at this early age in weight and in body fat. So we're really we're focusing on exercise in this, but as well as a diet intervention. So we're going to start introducing different diets, including an anti-inflammatory diet, to see if that may have a similar effect to exercise. And one thing we're really interested in is if we put these mice on the anti-inflammatory diet without exercise and then give them a wheel, will that improve their running distance? So if we take away that inflammation through diet, will they run as much as a naive mouse? Okay, I have no idea what time it is, by the way. Am I? Okay. So one of the things we're interested in is moving away from this early life stress paradigm. It's very interesting. It gives us robust results. It's a pain. It is, I don't mean that in a punny way. It takes a lot of time. It's very expensive. It's fraught with peril in that we've had mice deliver en route. Okay, those mice are done. We've had mice that show up, we think are pregnant, and they're just fat. We just ordered 8 mice in last week, and we got 3 litters out of them, and that's not usable. So it's a lot of time, it's a lot of energy, and we need to expand, because that's just a lot of work. So we've started looking into an adult stress paradigm, and this is done in conjunction with Doug by a student that we're co-mentoring / co-parenting, Yen Wu. And so one of the interesting things about using black 6 mice, everybody uses them, but they're really low anxiety. They're really hard to agitate. So they already have this low anxiety phenotype. They're hard to inflame. They're just really resistant to a lot of the things that we want to do as people interested in pain. So they're way down here. But there's another strain, AJ, which are really high, and they exhibit increase in pain. Doug's used these in his diabetic neuropathy studies. They develop painful diabetic neuropathy as opposed to black 6, which develop insensate diabetic neuropathy. And they've shown to have an increased activation of the HPA axis. So we're like, oh, well, let's trust these guys out. So now we work on these cute little furry white mice. And what Yin has developed is this foot shock paradigm, which is modeled after work from UAB that Tim Ness and Meredith Robbins did, looking at the interaction of CRF receptors in the spinal cord. So we order in these mice around 12 weeks of age. We get baseline measurements on them, and then he does a 10-day randomized foot shock. So we have this plate that elicits a foot shock. So he puts them in, and for 15 minutes they get 30 randomized shocks. And so you either get the shocks or it's a sham exposure, so that we have an appropriate control. And what he's done is he's measured sucrose preference over this time, and then we're measuring after the shock. So far we've only done the 24-hour time point, but he's also working on a 30-day to see how chronic the effects are. So one of the first things we did, what everybody does, is to look at hind paw responses. So this is looking at mechanical withdrawal thresholds to those von Frey monofilaments. So at baseline, this is prior to any exposure. Our two groups are essentially identical. After sham exposure, and this is 24 hours after the 10th shock, we see no difference, but we see a significant decrease in the animals that received the foot shock. So they are different from their baseline measurements as well as from the sham group. We also did bladder distension. We don't have baseline measurements, but this is 24 hours after, and we saw a significant increase in the VMR to bladder distension in the animals that were shocked. Interestingly, the shock appeared to make our mice depressed. So this is, you probably can notice like the black 6 mice, and this is something that Yen pointed out to me, they're somewhere around 80%. They really like the sucrose. They don't have anxiety, they don't have depression. They're like, woohoo! They're super psyched about the world, the black 6. But the AJs already start out around 60%. And it drops to around 50% during this shock paradigm. Whereas in the sham animals, they increase just a little bit or stay steady. So it results in significant anhedonia during this time. And then looking at activation of the HPA axis, we do see a significant increase in serum corticosterone in these animals 24 hours after that shock. And we looked at mast cell degranulation, and in hindsight, I was like, oh, duh, okay. So if we think these sham animals are like our maternally separated animals, I should have anticipated that they were at 80%. So this is showing that they also have this already ongoing peripheral inflammation, this kind of systemic activation through the HPA axis, and then shocking them had no effect. Okay, so grand conclusions. So our model of neonatal maternal separation, hopefully I've convinced you that we have this urogenital specific visceral hypersensitivity. So vaginal distension, bladder, perigenital activity, all of the responses show hypersensitivity. They do express anhedonia. They have reduced hippocampal regulatory gene expression that is involved in regulating the HPA axis, and we're investigating potential metabolic syndrome. Early exercise intervention can reverse most of these effects. Water avoidance stress can exacerbate most of them as well. Foot shock exposure in these adult AJ mice can also result in hind paw bladder sensitivity, anhedonia, and increased serum corticosterone. Some of the future directions we're interested in, like I mentioned, the effect of the diet on the NMS outcomes, using a high-fat or high-sucrose diet versus using anti-inflammatory. Can we make them even more American by giving them a McDonald's diet on top of their stress and their sedentary lifestyles? And can we reverse that with a healthy diet? The chronic effects of the foot shock. So we've shown that 24 hours after this paradigm, we see increases. How permanent is that? 30 days later, are they still hypersensitive, or does it go back to baseline? How much stress do we have to put them under to make it permanent? And we're really interested in getting into neuroimaging and seeing, basically I'm trying to make tiny humans, tiny furry humans that have all of these comorbid overlapping pain disorders, and how much do they look like the patients that are going through the MAP program and show changes in gray matter volume and functional connectivity and all these important brain regions. So that's kind of a future direction. And finally, I didn't do most of this myself. So I have an excellent lab full of students and trainees that have done all of this work. I have excellent collaborators, very generous funding, and I will take any questions. Thank you. Rock chalk. Yeah.
So I've been thinking a little about this, and it looks to me like a lot of your neonatal stress and your adult stress are having the same pattern. think there's something unique about the neonatal period, or is it just stress and pain regardless of when that happens? It's a great talk, by the way.
Thanks. Well, they kind of are. I mean, the effects in the water avoidance stress in the naive animals is only for that one day. So it's not, there's no longevity to it. It's kind of that acute increase. And it was different depending on, so in the females it increased colonic, and in the males it increased perigenital. So there are sex differences, but there is something unique about that neonatal time period that makes them more susceptible. We had another study that I didn't get a chance to talk about. So when I was at Pittsburgh, I was doing neonatal colon insult and looking at the long-term effects and how it increases colorectal sensitivity. So another part of my COBRI project was to do it vaginally. So we used zymosan, which is a component of the yeast cell wall, gave that intravaginally during the neonatal period, and then looked at vaginal sensitivity in adulthoods, and it did increase it. And the reviewers, for some crazy reason, wanted me to do it in adults. So we took adult mice, put zymosan intravaginally, and then measured it the same time period later, and it had no effect. So there is something about that neonatal period that makes them more susceptible, primes them, if you will. Yeah.
So I had another question in that vein. You were showing what looked like a dosage effect in the neonatal stress paradigm. And I was wondering, I seem to recall that when you were doing chronic mustard oil as a neonatal insult, you did a couple of time points, and it looked like there might actually be a critical period.
So we did,
So
yeah.
would you, I mean, is there a way to tell the difference between whether what you're seeing is a dosage effect or maybe a critical period during that time course that you're missing the one day?
There is a way, and it would take a lot of work. And I get asked that question all the time. So if we just separated in that third week, would we see it? So the thing about the third week, and there's not a lot of neurogenesis going on then, but there's a lot of synaptic plasticity, synaptic formation in the hippocampus during that week. And so we think we're really messing with that. And that's what's driving it, since that's really what we're seeing as far as central changes in the hippocampus. That's what we really think. Whether or not we would see that if we omitted the 2 weeks before that, I don't know. And all the somatic studies, like Mariah Fitzgerald stuff, it's that first week. So if you want to mess with the hind paw, Mike could probably speak to this better than I can, you need to hit it even earlier. So viscerally, second week is fine. You know, people have looked at 14 to 16, you still get an effect. So there's even a different window depending on the structure that you're insulting. Yeah?
I know anomalies always are aggravating, but sometimes they're interesting. And I don't have a good enough handle on all the different paradigms that you use. But sometimes when the females are more, and one of the females are more desensitized to pain, and I forgot which outcome we were doing, which was somewhat surprising when the males were not desensitized. And in the cardiac world, we found that, whatever phenotype we're trying to do, cause hypertrophy or whatever, the males would always have a much more robust phenotype than females. And if you step back and go, wait a minute, females, because of the reproductive challenges you have cardiovascularly, have a much more robust change to perturbations than males. I'm curious, is there any reproductive aspect that can differentiate your males and females? And you saw your males are very sensitive to their genital stuff. Some of the bladder distension for the females are less sensitive? I don't know, I'm making it up.
You can imagine the behavioral tests that we've, brainstormed in the lab to get at that, and the, creative titles we could give them. We haven't looked at it. There have been studies that have shown hereditariness of this, particularly in males. I can tell you that the males are harder to fix. So we've done different intervention paradigms with the exercise. So the one that works the best is starting at 4 weeks. So if you start them at 4 weeks, you pretty much can correct most of what we see. If you start at 8 weeks, it's not as effective. The females bounce back for the most part. We can correct them. The males could care less that they're running. They are still sensitive. They're still completely messed up. And it matches what's known in the human literature, that especially little white males do horribly after early life stress. So if they're born premature, they're less likely to thrive. It's an ethnic sex issue. So males are more susceptible to early life stress than females. I mean, girl power and all, but I don't know why that. We haven't really delved too far into it.
And to follow up a little bit, now I'm heart and diabetes, so that's going to make you do this, but no matter what we did, sucrose feeding and so all these other things, the males presented with a phenotype much earlier than the females. So that's sort of on the other end of things. The females are less susceptible to interventions. So I was curious if some of your male data maybe you need to look at an earlier time point
At an earlier time point?
than the female.
Yeah.
If you have the same time points, the males and females, the males may have already presented.
Yeah, I do know in the females, the phenotype just grows stronger with age. They become more sensitive. Our biggest responders for the visceral motor response are the oldest. I mean, it just gets more and more robust. So with the migraine stuff, that's why they were 10 months old, because the phenotype it deepens and blooms with age. It's like a fine wine, I guess. The outstanding condition. So it could be, but I do know that the males need a sooner. They need to be intervened with sooner. Yeah.
So kind of going off that, and maybe you mentioned this, and I just didn't hear it, but when you mentioned that you had that sex-specific effect, was this in prepubescent mice?
No, they're all adults. Yeah. The youngest we can do any sort of distensions is 8 weeks. So we can't really test any sooner than that, and they're adults at that point. So we haven't really gone much younger than that. Yeah.
Just curious, was your maternal separation, do you follow up to their core level with the mice?
Yeah, it's higher.
Is it like a throughout, or there's a, up and down?
I mean, it's changed with the cohorts. We do see variability between. So each group that we order in of the pregnant moms, half of them are separated and half of them are not. So I always have internal controls for environmental things we can't control. And it has changed, but overall, there's been a significant increase in the serum cort levels in those mice. Mm-hmm.
Because one of your data said it was in the after the water stress, and
Uh-huh.
there was a decrease and an increase, and day 8 or something after the stress. Does that make anything?
Yeah.
The rebound.
Actually, after water avoidance stress, the cort levels drop.
Right.
I didn't show that, but they drop over time.
Is that correlated with the core levels? So by,
We didn't measure both in the same animals,
okay.
so I couldn't answer that.
And then another, just curiosity, for the foot shock study you did,
Uh-huh.
you have in the follow-up long term, right? So in the past, when I did a whole week of a foot shock, it's a random foot shock, what I found was if I follow up for 4 weeks, the peak time I see the increase of the cord is the first week, and then they actually develop an adaptation. We measure the cytokines, later on you see more anti-inflammatory or inflammatory. So I don't know if that might be something happening in your model too, if you follow up.
What animals do you use?
Which was about C.
Okay. Okay. Yeah, it would be interesting. I'm not sure what we'll see. Since it's already a strain that's susceptible to stress and anxiety in the first place, I mean, I think if we did it in a black 6, they would habituate to it. Like, I don't think we would see anything because after a while, because we've done the water avoidance stress where you do it for 7 days in a row, and the mice habituate. They don't care. Like, it has to be acute, or else they're like, whatever.
Right.
We'll swim.
When I compared adult C and the B6, adult C has a higher core level after stress.
Okay.
So,
Good to know. Thanks. I'm going to skip you.
Thinking about the first thing that we were talking about, the neonates. So I've had puppies. Well, my friend did. Actually, dogs, puppies. The dog had puppies. So the dog again. And going
Mhm.
on. So, and in the milk, I can imagine there would be growth hormones and stuff, or at least some nutritional factor that may contribute to urogenital development, but also the licking.
Mm-hmm.
I don't know about rodents. I should know, but I don't know about rodents. But in dogs, at least, they don't void
Right.
until they're actively stimulated by their mother's
Same with rodents.
So, I suppose the question is there any, at baseline, after neonatal separation, are there any gross urogenital differences? I suppose, I'm sure you've probably looked, right?
We haven't, actually. That's a good suggestion. That has come up when I thought about it. And the earlier study that Derek brought up, that's a question. I mean, I was putting mustard oil. It's such a mean thing to do. But I would think that would deter the mom from licking, because that's not pleasant. Nobody wants that on their tongue.
You could use that as an experiment.
Yeah. So they are going 3 hours without that stimulation. It is during the light cycle, so they wouldn't theoretically be getting a lot of stimulation, but they would have that contact with the mother. And there have been studies, we have not looked at it, but there are studies that have looked at maternal behavior and arched back nursing versus curved, and how those interactions relay into the development of the pup's later life.
And I had a kind of an extra question, almost a weird one. Like, do you think there's a phylogenetic reason for this that would influence the early life stress would influence mate choice in later life? So that is it possible that there's an actual mechanism to influence mate choice? So if you're, no?
I have no idea. What do you hypothesize?
Well, no, I mean, it might be to do with the makeup of the colony, or in the humans, the tribe, so that it's a bit weird to talk about with you.
It makes you codependent? Like you went through this early life stress and now you're extra needy and need a codependent spouse? I don't know.
No, yeah, maybe I'll stop right there.
Okay.
No, don't stop, I'm enjoying it.
I know.
Well, there were some real personality differences in the famine studies,
Yeah.
like the genetic study.
Yeah. Well, there was a woman came and gave a talk to our dietetics group, and it was very interesting. So she was looking at stress responses in vaccinations, and it was like it was some island with a horrible caste system where indigenous people are very poor, and then the rich people came in and they're very wealthy. And she was correlating socioeconomic status with stress, and made the assumption that if you're of low socioeconomic status, you have more stress, and your babies are going to react more during their vaccinations. And she saw the opposite, and I was like, yeah, because they're likely wearing their babies, they have a greater social structure. Actually, these are better families to grow up in because there's an actual village taking care of everyone, and they're more close-knit. And then the wealthy babies that were probably like my kids and left in a bouncy chair while mom worked on her grant applications had a greater response to the vaccinations. And I was like, well, that makes, if you think about it, it makes total sense, because there's going to be more kangaroo care and baby wearing. And she was like, oh, I was like, oh, okay. So that kind of blew that out of the water, which was fun. Yeah.
Two things. It sounds like it's just solved the problem. All you have to do it for early life steps, make them run around.
Yeah.
Like we used to.
Yeah.
And the second point is, if she sat up closer, she would have asked you. She's had a question in the back.
Oh, sorry.
That's okay. So I don't really have an experimental outcome in mind, but in your hollow organ distension protocols, have you guys looked at repetitive pressure stimulations rather than escalating pressure distensions?
No, we do give each pressure 3 times, so it's in triplicate, but we haven't. It does cause a stress response. So it's, as you would imagine, it's stressful to be shoved in a little tube and have your organs distended when you're a mouse and you have no idea what's going on. So it is a stressful event in itself, and we do see changes following it, but we haven't looked at at repeated of the same pressure. Yes.
So I have a question about your BDNF as well.
Uh-huh.
Could you put up that slide?
Sure.
So I'm really curious about BDNF effect on hippocampus.
Uh-huh.
And I'm trying to make sure I got this right. Did you look at dendritic branching at all?
No, we haven't looked histologically yet. No, but Isabella's going to do BRDU staining just to look at, to see if there is any neurogenesis.
So, okay. So, but there is also, so there are some effects of BDNF on memory. There's some studies on memory and hippocampal sprouting. And so I guess and also I guess exercise on BDNF and BDNF-induced sprouting. So, but I guess, so I was thinking that you had a suppression of BDNF with stress, but that's not what you're seeing. You're just seeing an increase with,
Just an increase,
with
yeah.
exercise.
And the males show this too. I just don't have a slide of it, but they show an increase in BDNF as well.
So there are also cognitive effects of long-term stress.
Mm-hmm.
Would you, I guess, so that's what I was thinking about with this result. But would you suspect that you would see that could be related to impact on hippocampal BDNF, or that that's just a completely different system?
What? The cognitive effects?
Yeah. The cognitive effects with stress would have anything to do with regulation of BDNF.
I don't know. I mean, something is obviously affecting the ability of BDNF to be increased. I mean, because we see it selectively almost in, there could be.
I mean, in a way, this almost seems like the reverse. Because your maximum BDNF.
You're being disagreed with.
What?
You're being disagreed
I
with.
never disagree with Derek.
Okay. I do. It's fun.
No, I routinely disagree with you. No, I think your observation through the BDNF is actually consistent with other reports in the exercise literature.
Yeah. Yeah.
BDNF across different areas of the brain. But building off of what Derek was saying, have you actually tried to look at any social interaction studies?
We tinkered with it a little bit in the beginning to try to get a good anxiety behavior. So we did do like the novel, where you put a novel mouse in with them and see how much they freak out, essentially. And we have toyed with doing where you put the rat in the middle and then they see how much we've tried different anxiety behaviors and we haven't really seen anything there. But we haven't, again, getting at the reproductive behaviors. We've thought about it and usually it's just to laugh. But yeah, we haven't too much.
I was also thinking about that in relation with your nutrition studies.
Mm-hmm.
Do male rodents mark
Yeah.
territory? Because I'm thinking with your differences, maybe it's more in relation to that and not in sensory difference.
Yeah, no, that's a really good point because we have done more of the cystometry we've done with the males. And so Isabella she did a lot of cystometry in her males and we saw the opposite. So we saw we saw urine retention, higher pressures for release. It was exactly the opposite. So everything we saw in the micturition patterning, where they were going more, smaller voids, all of that was completely the opposite in cystometry. So if you actively fill the bladder and measure how it's voiding, it was almost had a bladder retention effect or urine retention effect. And then in the more behavioral study, it was increased. So suggesting, right, so we do think that gets at a really interesting question of how much of this is behavioral, prey behavior, prey species behavior.
I was thinking you might be able to
Tease it out.
two groups,
Mm-hmm. Mm-hmm.
dominant versus the
Yeah. We have noticed that there's not a lot of fighting. So they are pair caged, and the separateds, we've never had to take them apart, and we just had to separate some naives that were apparently fighting over their wheel. So it does tend to, we always say that, so the way I anthropomorphize my mice, The males just seem very depressed and withdrawn because they aren't, like they just have this kind of, and the female mice, I picture them with like a martini and a cigarette, like the big long cigarette holders, and just kind of melting against the wall, catastrophizing all over the place. That's kind of how I picture the two differences in how we see. And that, it definitely could be like adding that stress in, that water avoidance stress really could make it so that they don't want to mark.
Right.
Yeah. Yeah. Okay, thanks guys. Great questions.